Our final presentation of the evening is Wolf Biosciences. Please join me in welcoming them.
Thanks. All right, my name is Adam Young, and I'm the founder of Wolf Biosciences, a company that's pioneering new biomaterials for adipose or fat regeneration. Our lead product, AdiV, is an injectable gel designed from human liposuction fat that actually stimulates new fat growth at the site of injection. Now, I know it seems counterintuitive to offer products to help people gain fat. However, there are several patients that would actually benefit from this type of a strategy. So what I'm referring to here are soft tissue filler procedures in which a material is placed subcutaneously or beneath the skin in order to fill an adipose void or deficit. So this might be the case for a woman with breast cancer who's had a tumor removed and now she has a patient-specific void that now needs to be filled to replace that tumor. This could also be the case with HIV patients suffering from lipoatrophy, or even pediatric patients like this girl here who's suffering from Perry-Romberg syndrome, in which she is unable to produce adipose tissue on one half of her face. Now, what these patients represent is that our product, AdiV, by stimulating natural adipose regeneration, is truly a platform technology because we can treat a variety of adipose deficits as well as several secondary indications such as bladder incontinence, gastroesophageal reflux, and even cosmetic wrinkle filling. So while our passion is helping patients like this girl here, the cosmetic wrinkle filler market is enormous. And in fact, the developmental pathway for designing a product to treat this patient here is very similar to the pathway to design a cosmetic wrinkle filler here. So what this means is we can take advantage of the easier path to clinical trials with cosmetics in order to stimulate faster investor revenues that can then be used to not only support our company, but also support the development of tissue engineering products for these orphan-type diseases. So if we take a look at the cosmetic fillers market, you'll notice that there are 2 million soft tissue filler procedures performed in the United States every year. This represents $2 billion in annual expenditures, and that's only 40% of the international market. So what this means is that actually, in fact, this market size has quadrupled in the past 5 years. So we have a strong growing market. And in fact, we predict the aging baby boomer population will continue to sustain this growth for at least the next decade. Taking a look at the competition, you'll notice there are actually several competitors within this field. So it may seem like a crowded marketplace to try to enter. But in fact, all of these products suffer from a variety of setbacks. Most notably is rapid degradation. Most of these products are going to be completely broken down by the body within 6 months. The products that have a more longer prevalence actually suffer from chronic inflammation, negative immune response, and even fibrous encapsulation, in which the body actually walls this material off with scar-like tissue. And in fact, none of these products actually stimulate natural adipose regeneration. So what this highlights is that there's a significant need for a durable product that's biocompatible, that's also going to integrate with the surrounding tissue and stimulate natural adipose regeneration, and therefore restore the function of that tissue, but also provide a long-lasting effect that these patients are desiring. And that's exactly the approach that we took with our product, AdiV. We have developed a patent-pending process to extract proteins and other extracellular matrix components from human liposuction fat tissue. So we start off by collecting lipoaspirate tissue from several plastic surgery clinics in the San Diego area. We extract these adipose-specific proteins and process them into a liquid formulation. This liquid formulation is then available at the plastic surgery clinic to be injected directly beneath the skin of that patient. But the beauty here is that AdiV has been designed that as it warms up to body temperature, it self-assembles into a soft hydrogel, much like when you place Jell-O into the fridge. What this means is that we have a 3-dimensional scaffold that's delivered through a needle that fills that patient's specific adipose void. But more importantly, since we're derived from human adipose tissue, we provide a template for natural adipose regeneration. And in fact, that's exactly what we've seen in our preclinical studies. We've been able to use non-dilutive grant funding to support all of our animal studies to date, in which we established the biocompatibility of AdiV, and we actually did demonstrate new fat formation, which you can see here. So here's the skin of our animal, here's some subcutaneous fat, and all of this is our AdiV gel, and right there is significant new adipose tissue that has been developed within our product. And I know it seems weird to get excited about making fat, but this is precisely where our competitive advantage comes into play. No other product on the market can stimulate adipose regeneration, and that's exactly what we saw with some of our clinical standards. We included Juvederm and Radiesse, which represent 90% of the soft tissue filler procedures in the United States. And in fact, there is absolutely zero adipose formation in either of these products. And in fact, here and here, you can see that fibrous encapsulation that's indicative of a negative immune response that I was talking about earlier. So not only do we have the only product capable of generating new adipose tissue, we also were able to produce new blood vessels within this gel. So we're providing nutrient supply for this newly developing tissue. So from here, we're going to initiate GLP pharmacology and toxicology studies that will further establish the biocompatibility of Ativi. And next, we're going to initiate a 5-patient first-in-human trial. Now, we've developed a unique protocol that will help facilitate this rapid progression into human trials. And following IRB approval, we can identify patients with a pre-scheduled abdominoplasty procedure. What these patients are people who have had a lap band or some other surgical procedure at which they now have excess skin flaps that are scheduled to be removed at some point in the future. So what we can do is, after gaining patient consent, we can inject Ativi beneath that skin that's scheduled to be removed, come back 1 to 3 months later, and collect detailed human histopathology data from this waste tissue at no further risk to the patient. And we expect to be able to do all of this within the next 2 years. So in 2 years, we are going to have early human clinical data that we can use to support follow-on financing. We've also developed a strategic alliance with Cytograph Tissue Engineering, which is a regenerative medicine company in the Bay Area that has actually already raised $25 million themselves to support ECM-based materials. But what's important about this partnership is that it gives us access to their GMP-certified manufacturing facility. It also gives us access to their regulatory and commercialization expertise. And these are going to be the 2 critical factors that help support our acceleration into these rapid human trials. Looking at our path forward, we've been able to use non-dilutive grant funding to support Ativi ideation, as well as our preclinical animal studies. Currently, we are seeking $350,000 to support GMP manufacturing of Ativi, as well as our GLP farm tox studies and this first-in-human study. So what this means is that by Q1 of 2015, we are going to have human clinical data for our product that we can leverage to secure follow-on financing that will support a broader phase 2 human trial, as well as our FDA submission. Looking at our business model, we envision Ativi as an allogeneic, off-the-shelf product that's available at the clinic. In other words, we sell this product to the plastic surgeon, who in turn sells it to the patient. Currently, we can produce 400 doses of Ativi from 1 lot of donor tissue. We also expect a sale price of $250 per dose. This is the average cost of an injectable filler on the market today. So if we only gain 1% market share, that's 20,000 doses sold every year and $5 million in annual revenue. And with moderate estimations, we predict we can gain at least 5% of a market share by 2023, representing $30 to $50 million in generated revenue. We've assembled a diverse team at Wolf Bio, including myself, who designed Ativi as part of my graduate thesis. We also have Dr. Karen Chrisman, who co-founded another university startup and has already had experience raising $3 million in SBIR funding for that company. We also have Dr. McAllister, who brings critical commercialization and regulatory experience to our team. And he's also been managing and running another biomaterials company for the past 13 years, bringing critical management experience to our team. And finally, Dr. Wallace, who's the chief of plastic surgery at UCSD, who's going to help critically with the design and execution of our human trials. So hopefully I've convinced you through this talk that at Wolf Bio, we offer the only product capable of stimulating adipose regeneration. We can also produce Ativi at or below the manufacturing costs of all of our competitors, which will allow us to maintain a healthy 40 to 50% operating margin. We've also secured research and manufacturing capabilities at an early stage, and our rapid progression to first-in-human trials will provide a major valuation inflection in 2 years that will help support, that we can use to leverage for follow-on financing. And finally, most soft tissue filler procedures are paid for by the patient out of pocket. What this means is not only do we have an amazing regenerative product, but we also avoid the hurdle of insurance reimbursement and can therefore provide a faster return on your investment. And with that, I'd like to thank you for giving me the opportunity to pitch tonight. I'd be happy to take any questions.
we have any questions or comments from our judges? Sure.
I'm curious about the IP, the intellectual property protection.
Mm-hmm.
I mean, you're purifying out existing mediators from from the liposuction suction procedures.
Correct.
How does that stop from somebody else going to the plastic surgeon next door, getting their extra fat, and then running a similar manufacturing process?
Yeah, so our IP covers the production of extracting these ECM components from human liposuction tissue and generating a product from that downstream. And that patent was filed in 2009, so we're currently in process on that right now. Hopefully that helps.
So what is the mechanism by which the adipose regeneration is controlled in the body? In other words, how do you know once you put it there, it's either going to stay there or you don't end up with some...
That's a great question. Yeah, we don't want to stimulate too much adipose generation, huh? Yeah, so the material itself will go in, it forms a bolus in the void that you put it in, and that's exactly where it stays. We haven't seen it migrate anywhere else from that. And the material will slowly break down as this adipose tissue infiltrates. So once the material is gone, it's going to stop encouraging new fat formation because the material is basically providing a template for the body's natural progenitor cells to migrate in and then produce more adipose tissue. So as that material breaks down, we're actually regenerating more tissue to fill its space and not causing a significant total upheaval of adipose regulation.
So you're using other people's fat cells. Is there no concern about the body's immune response to the material that you're injecting?
Correct, that's a great question. And I kind of glossed over, that's part of the science of this, is we're doing a process called decellularization. So I take the human fat and remove all of the cellular components that could potentially be an immunogenic component of the tissue. So this has been seen, there are several products that do this decellularization process, predominantly for wound healing. Cardiovascular applications are coming up. And what we've seen is by removing that cellular content, if you get below a certain threshold of remnant DNA, there's really a negligible immune response, and it turns into a more beneficial vascularization and regeneration type response.
Are there any further questions for Wolf Bio?
So after the round A and you do your first in humans, have you thought about how big the next clinical would likely be for FDA submission?
We're estimating around $2.5 million to run about a 200-person human trial to get that data up, and then we'll be doing 510K submission for the FDA. And since we're a low risk profile and being subcutaneous, we actually only need IRB approval to initiate those initial human studies as opposed to FDA approval.
All right, with that, let's thank Wolf Bio.
Thank you, guys.